Okay, good evening, everybody. My name is Autumn Anglin. For those of you who don't know me, I'm an artist, a graphic designer, and photographer. I live on a 1-acre farm in South Salem, Oregon, in the heart of the Pacific Northwest, and I live in a region known as the Willamette Valley. It's a temperate forest, and we have two mountain ranges on either side of us, the Coastal Range and the Cascades. Hold on, give me 1 second. I'm going to start presentation. All right. So I just finished a month-long interactive art show called The Art of Fungi, where I hung 25 photographic prints with scannable QR codes that people could scan with their smartphones and learn more about fungi. This hung for the month of January at the Infinity Room in Salem, Oregon. I'm writing a book right now called The Year of the Mushroom, and this presentation is a sample of what I'm writing about. It will be a field guide for the Willamette Valley specifically. I'm on the board of the Inglewood Forest Festival, a community art show in Salem. I'm also on the board at Librarians with Spines, an anti-racist publication that's publishing its 3rd volume in a few months. I own 2 businesses, Autumn Steam Ceramics and Grey Girl Graphics, where I sell handmade ceramics and do freelance book design, logo design, web design, and graphic design. I'm a member of WVMS and have been since 2016, and I've been their treasurer since 2017. I created the WVMS study group in 2019 with the help of Henry, Diana, Jordan, Jeannie, Beth, and Patrick. I'm still learning and by no means an expert, but I'm excited to share what I've observed and photographed over this past year. Since the creation of the study group, I have created a year-long self-paced introduction to mycology class for WVMS members that I teach students how to identify fungi in the field. We also started an iNaturalist WVMS funga project where we have shared our fungi observations and joined forces with the Fundus project, which is NAMA's new citizen science project. Our study group has gone out to observe fungi every couple weeks for 1 year and have seen some amazing specimens and even some rare fungi. My favorite mushrooms to find are the little black cup fungi, and I purposely seek them out every time I foray. I have a few mushrooms getting DNA sequenced right now through Fundus, and I should have those reports back at the end of the month. So in this year, I have learned so much, but been wrong about almost everything and humbled again and again. So tonight I want to talk to you about the seasonality of mushrooms, the mushroom growth cycle, precipitation, when to find mushrooms and in what habitat, how the climate crisis is going to affect our fungi, and vocabulary for observation. And then I want to show you some examples of the mushrooms that I have observed in each season. This presentation is really long, and so I'm going to give us a 10-minute intermission, like I said before, but I do hope that you will stick with me. All right, so let's get going. So what is a season? So according to the National Weather Service, we all know that the Earth makes a complete revolution around the sun every 365 days, following an orbit in elliptical shape. This means that the distance between the Earth and sun, which is 93 million miles on average, varies throughout the year. So during the first week in January, the Earth is about 1.6 million miles closer to the sun than is referred to as the perihelion. The aphelion, or the point at which the Earth is about 1.6 million miles further away from the sun, usually occurs during the first week of July. This fact may sound counterintuitive to what we know about the seasons in the Northern Hemisphere, but actually the difference is not significant in terms of climate, and it's not the reason we have seasons. Seasons are caused by the fact that the Earth is tilted on its axis by 23.5 degrees. The tilt's orientation with respect to space does not change during the year. Thus, the Northern Hemisphere is tilted towards the sun in June and away from it in December, as you can see on those graphics. So winter is from the solstice to the equinox, which is about December 21st to March 20th. The winter solstice marks the shortest day and the longest night of the year in the Northern Hemisphere, and it occurs when the sun is directly over the Tropic of Capricorn. Spring is from the equinox to the solstice, which is about March 20th to June 21st. There are only 2 times of year when the Earth's axis is tilted neither towards nor away from the sun, resulting in the nearly equal amounts of daylight and darkness on all latitudes. These events are referred to as equinoxes. The word equinox is derived from 2 Latin words, equus, meaning equal, and nox, meaning night. Summer is from the solstice to the equinox, which is about June 21st to September 22nd. The summer solstice occurs at the moment the Earth's tilt is towards from the sun at its maximum. So the sun is at its highest point in the sky, and this is the longest day of the year. Autumn is from the equinox to the solstice, which is September 22nd to December 21st. So the autumnal equinox is the beginning of when the nights become longer than the days. All of what we consider seasons in reference to the calendar or solstice and equinox is not what we're referring to when we're talking about mushroom seasons. Like the growing seasons of crops and plants, mushrooms are tied to temperature, precipitation, elevation, substrate, and habitat. The calendar seasons are more of a general way to think about it when you like how to find a mushroom. But it takes a lot more effort to observe your own microclimates to know when a mushroom will fruit. So according to Maud Saleh-Senussi, the research officer at the Mycology and Pathology branch of the Forest Biodiversity Division in Malaysia, there are 2 stages of growth before the mushroom completes its life cycle. The first stage takes place underground, requires weeks to complete, and involves the formation of the prototype mushroom, or the pinning stage. This is the period where mushrooms live in the form of tiny buttons in the mycelial layer and are hidden from view, usually under the substrate like duff or soil, or under the bark of a dead tree. These buttons are tight-bound structures similar to the compressed, like dried sponge that can expand rapidly when it's exposed to enough water. The second stage of growth begins when the water intake and moisture level is high enough. So according to the Mycological Society of Marin County, usually an accumulated 2 inches of water will start a flush. The mycelium draw in huge amounts of water, expanding the button and spearheading the fruiting of the mushroom. Smaller mushrooms can grow within a day, while medium and larger mushrooms require about 3 to 4 days, provided they have a steady supply of moisture available. Different groups of mushrooms might take on different growth habitats. Some take only a day, while others, will last a few days after their maturity. For those of you who have grown mushrooms in your kitchens, you know that some mushrooms need the right light, the right moisture level, oxygen levels to produce a fruiting. The same set of conditions need to be right for wild mushrooms to fruit in the forest. Okay. Here is a map of the area where I do most of my mushroom hunting. So I want to show you the differences in precipitation in Salem, or in the valley here, and in the western facing slopes of the Cascades, and in the coastal range. So this is Salem, Oregon, home of the Willamette Valley Mushroom Society. And we are in the Willamette Valley, situated right between two mountain ranges, with the Pacific Ocean and the Gulf of Alaska supplying us with seasonal variations in precipitation and weather, including lots of rain from this temperate rainforest from October to March. And then the rain sort of in precipitation declines after March, and we usually have pretty dry summers. So in Salem, we get about 45 inches of rain per year. So on this graph, you can see the average temperatures, the blue and red lines, and the average precipitation, the light blue bar graphs there, per month. So I highlighted the 2-inch mark here of precipitation because that's at the level of accumulation needed for some mushrooms to begin fruiting. Of course, there's other things that are needed for the mushrooms to fruit too, like temperature and habitat and elevation. But precipitation is a good place to start when you're trying to figure out when a good time to start foraging or studying would be. So I want you to specifically look at September and October. These are the months that start or delay our wonderful fall mushroom season. The fall mushroom season in the Pacific Northwest is the best, with the most variety and the most interesting things. At the Willamette Valley Mushroom Society, we are always especially excited to kick off the fall season. In September, on average, Salem does not accumulate enough precipitation to start the mushroom season. But you can see by October, we should have accumulated 2 inches and beyond. So now let's look at the western facing Cascades. These forests are full of Douglas fir, hemlock, fir, some deciduous trees like alder, maple, and oak. There is very little old growth though, and a lot of it is made up of plantations, monocrops in various ages. This is a little town called Hydnum, which I chose because of the mid elevation at about 1,700 feet. And this town has weather data, so it was important. This is usually the farthest east I would go to mushroom hunt, and but this past year was devastated by a wildfire that has shut down access to millions of acres that have burned. Some of the photos I've taken for this presentation were habitats and fungi that are now destroyed. The first foray I ever went on was led by Jim Shepke in this area, and it was in this Santiam Canyon corridor where I first learned how to hunt for chanterelles and learned how valuable rain gear was. So Idana gets about 56 inches of rain per year. And if you look at the pink line, you can see in September, there's usually an accumulation of just over 2 inches. By October, there is more than enough to get the mushroom season started. This area does get snow, but as you can see, it's kind of rare in October. Now we're going to look at the coastal range. This is where I do most of my mushroom hunting because it is so good all year round. So I've gathered weather data from Otis because it's near where I do a lot of mushroom hunting in the Sislaw National Forest. And Otis's elevation is only 56 feet, but I have traveled up to about 2,000 feet at the coastal range to foray. Otis has about 79 inches of precipitation per year, and you can see September seems to have enough moisture to start a fall mushroom season off earlier than Salem does. On the coastal range, in the slopes that face the Pacific Ocean, you're going to find heavy rainfall and lots of fog drip. This keeps the forest moist and the mushrooms fruit all year round. So from Yachats to Tillamook, the coast gets about 85 to 95 inches of precipitation per year. You can see there isn't a lot of time where the coastal range doesn't have an accumulation of precipitation. So much rain there. Okay. So when we try and predict places to go foraging, keep in mind that Salem gets far less precipitation than the close by mountain ranges. But remember, it takes some other factors too to start seeing fruitings, like the 3-week pinning stage before you see a growth of the big mushrooms. So here's a graph of the precipitation by day in the month of September for 2007, 2011, 2016, and 2019 in Salem. Now, mushrooms need an accumulation of a couple inches within a week to 10 days, and then 3 weeks from then to fruit. So if you look at the red dots, that was 2011. There is an accumulation of less than a half an inch of rain in all of September. So the fall mushroom season was most likely delayed a few weeks. If you look at the green dots, you're going to see 2019 had an accumulation of a couple inches of rain by mid-September, but we need to keep that moisture up to have a consistent season. So now if you look at October, same years, you can see that 2019, the green dots, they sort of fell short in October, but 2016, those yellow dots really started dumping out the moisture. This was the year of WVMS's first mushroom show, and it was so awesome. It was at this show that I actually became a member. It's hard to predict the exact time of the mushroom season, and it varies from year to year. You can see the amount of rain Salem got in 2007, 2011, 2016, and 2019. As I recall, the beginning of the fall mushroom season in 2019 began in late September. Our mushroom society had gone on a camping trip together in late August at the coastal range, and we found an amazing flush of lobster mushrooms, or Hypomyces lactiflorum, and then began our regular forays in mid-September. Alternatively, 2020 had a very dry September that resulted in a later beginning of our fall mushroom season, and the drought led to a horrible wildfire season. You can see here that if you need an accumulated 2 inches of rain plus about 2 to 3 weeks of growth, September did not offer the right conditions in Salem, but October was getting much closer. So now I want to talk a little bit about habitat. In the Pacific Northwest, and especially in the forests near us, we have Tsuga heteropylla, or Western hemlock. These trees are shade tolerant. They have the highest growth rate ever recorded in a temperate zone. And the Western hemlock also has the densest canopy of any tree species in the West. There are very few understory plants that can grow under it, especially in about 30 to 80-year-old forests. Western hemlock will grow on sites with significant organic content where the soil, but won't grow in floodplains. So when trying to ID this tree, look for fallen hemlock cones. They're pretty tiny, sort of opened up a bit. You can see a branch here in the picture. So you're gonna wanna look for short, like flat, blunt needles on sort of gracefully down sweeping branches. The other major tree you're going to find is Pseudotsuga menziesii, or the Douglas fir. So the Latin name pseudo means false, And Tsuga means hemlock, so this is a false hemlock. The species was first described by naturalist surgeon Dr. Archibald Menzies, and its common name is named after Northwest explorer botanist David Douglas. These trees only showed up in our region about 7,000 years ago. Under natural conditions, Douglas fir does establish itself primarily after fires on wetter sites, and they can live for 1,000 years. This is why we have some old growth forests in Douglas fir. So you can identify Douglas fir by its distinct cones. They're about 5 to 10 centimeters long, or like 3 inches-ish, and they feature a 3-forked, like papery protrusion there, see by my nail, that come out from its rounded scales. So certain mushrooms love these cones, and I'll talk about those a little later. These trees can get about 230 feet tall. The needles are pointed and soft and sort of spirally arranged. Diana taught me how to look at the branching on all the conifer trees out in the forest. And Douglas fir have some branching that are kind of sporadic and look scattered. There's many more trees and plants in our area, but these two make up the most habitat that I hunt in, and I just don't have time to go over all of them right now. Okay, so here you can see some of the beautiful forests that I look for mushrooms in. These forests offer connected ecosystems of mycorrhizal fungi that depend on specific trees and plants to survive. The Greek word myco means fungus, and the word rhiza means roots, which refers to the mutualistic relationship of the hyphae and roots. These forests and almost everywhere else also have the right conditions for saprobic fungi as well. Those break down matter into usable carbon and nitrogen for plants to use. The forests are a treasure, but they are changing. So as the climate crisis progresses, we're going to see more extreme weather events. We saw evidence of this in 2020 with the wildfires in September and the smoke that turned the skies dark red for days here in Salem. And in that devastating ice storm of 2021 in February, where the ice coated the trees and broke most of the deciduous trees, just devastating our landscape and impacting our communities. Continued global warming will lead to accelerated sea level rise, ocean acidification, declines in snowpack, and the loss of glaciers in the Pacific Northwest, with the negative consequences for the ecosystems, communities, and industries that are critical to local cultures and livelihoods. The impacts of these deficits will not be distributed equally across countries, communities, and industries. And this will be an abrupt change, not a gradual one. We're going to lose species. We're going to have different things going on with our global water supplies and total losses of ecosystems. Okay, so I'm going to exit my presentation because I want to show you this, what the effects have had on on this website, you'll see here this green outline here, and this was the fires that have that burned in the wildfires that burned in September last year. These are areas that I personally mushroom hunt in, and you can see the total devastation of communities, forest ecosystems, and possibly the loss of species. I have not been able to go back in and personally see the devastation because of the national forests are closed. but you can see the impact the wildfire has had on our beautiful forests. I just want to zoom in and show you. So this is Detroit Lake. This is the area that Jim took me on my first foray. And I'll just let this load up for a second here because it's kind of slow. But you can see the difference here, and all that brown is just everything that burned compared to what it was, you know, a year ago. Anyways, this map is on that set of links if you want to go, you know, search around and go play with it some more. I'm gonna go back to my presentation now. Okay, we're back. Okay, so now I want to talk a bit about the climate crisis. In my research for this presentation, I wanted to show how the climate crisis was and will impact fungi and their seasons. Unfortunately, fungi is not a priority for most institutions to study, so there's little information on that specific topic. Instead, I had to look at how the climate crisis is impacting the Pacific Northwest forests and our communities, and then draw my own conclusions based on what I already know about fungi. So in fact, according to Jessica Halofsky, who wrote a paper in the Forest Ecology and Management, the US has only been practicing climate change vulnerability assessments and adaptations for the past 10 years. It's very important to note that the climate crisis is real, and the environmental agencies and agriculture industries have all put out research on how to adapt to climate change. One of the ways the forest management is going to be doing it is by saving more seeds, planting hardier plants. And, you know, this is going to change the industry and the culture. Sustainable forage. So I just want to share with you what sustainable forest management is defined as the management that maintains and enhances the long-term health of the forest ecosystems for the benefit of all living things, while providing environmental, social, and cultural opportunities for present and future generations. To me, that's a huge ask, and you know, we all need to think about how we're going to help support that sustainable forest management. So what is the climate crisis? So according to the University of Washington College of Environment, in their climate impacts group, global temperatures spanning longer than a century indicate that the Earth has warmed by 1.5 degrees Fahrenheit on average between 1901 and 2012. The warmer temperatures are not limited to the atmosphere. The top 250 feet of ocean surface waters have also warmed by almost a degree from 1971 to 2009. And the warming trends are evident at nearly all depths of the ocean. The Pacific Northwest has warmed at about 1.3 degrees Fahrenheit between 1895 and 2011. With statistically significant warming occurring in all seasons. The warmer temperatures have increased the frequency of warmer extremes and mitigated cooler averages. In fact, the frost-free season, or the associated like growing season, has lengthened by 35 days from 1895 to 2011. Other indications of warmer regional temperatures are evident in the snow, ice, and stream flow trends. Spring snowpack fluctuates substantially from year to year, but declined overall in the Washington Cascades from the mid-20th century to 2006. This trend is due primarily to increasing regional temperature and reflects the influence of both climate variability and climate change. So natural variability can dominate over shorter timescales, but the resulting in an increase in spring snow accumulation in recent decades. But ultimately, the warmer temperatures are going to cause the snowpack to melt and melt sooner. So the decrease in snowpack will affect stream flow and water quality throughout the Pacific Northwest. And warmer temperatures will result in more winter precipitation falling as rain rather than snow, particularly in the mid-elevation basins where winter temperatures are near freezing. Those result will be less winter snow accumulation, higher winter stream flows, earlier spring melt, and earlier spring stream flow, and low summer stream flows in rivers that are dependent on snowmelt. So this graph shows the models of or predictions of global warming due to the greenhouse gas emitted globally. These are different projections, with the black line being our historical average. And the Pacific Northwest is projected to warm rapidly during the 21st century relative to the 20th century. The actual amount of warming occurs in the Pacific Northwest after about 2050, depends on the amount of greenhouse gases emitted globally in the coming decades. So what we do now matters for what is going to happen in just 40 years from now. So no matter what we do, though, we're on a trend for global warming, and the warming is projected to continue through the 21st century in the Pacific Northwest. So if you want to go play around with a climate model, this is a really simple climate model, and you can slide our emissions rate and see like how our CO2 concentration and temperatures will be affected. So this link is also in that packet I sent you earlier. So this is a way oversimplified model. There's so many facets to climate change, but you can get an idea of the impact of our emissions. Start at 0 with when you select your emissions rate, and see what would happen if you slide that over and do the projection. Our temperatures and everything will actually decrease, but that would mean humans would not be able to emit any more emissions, which I don't think is possible. And then if you put it at 6, which is sort of a goal for us, and that's 6 gigatons of carbon emissions. And then put it at 8.5, and that's sort of where we're at currently in the world. Okay, so just a month ago, some of the leading climate scientists in the Pacific Northwest had a conference about the drought and climate outlook. Most scientists are worried about water shortages, and this will impact mushroom seasons as we move through the 21st century. I would suggest you go look into these webinars if you're interested in climate change and adaptions. And I suggest you watch the webinars from this. It's drought.gov. They're, I think, listed under webinars or news and events or something like that. Also think about adding to your citizen science work and join COCORUS. This is a community collaboration rain, hail, and snow network, and submit your precipitation observations. So a lot of climate scientists actually need more weather data on microclimates, and they're asking that people put up these, the little tubes, like the weather precipitation things, and you go out, and even if you don't get rain, you upload your data to help them see what's happening. They need more data to be able to predict what's going to happen. So if you're interested, it's easy. You just sign up for an account, get one of their meters there, and upload your observations. So as we consider climate change, we should also think about adaptions and learning ways to adapt for mushroom hunting in our forests. But we should also be using fungi to help us adapt. Now, I was going to play a short video for you tonight, but this presentation got so long that I had to cut this out of my presentation. So I highly suggest you go watch this. There's a YouTube channel called I Contain Multitudes, and there's a specific video called Can Fungus Save Plants from Global Warming? It's like an 8-minute video. Just go watch it. It'll give you some hope after all of that doom and gloom. Another documentary I recommend is Kiss the Ground, and you can find it on Netflix. Okay, so when we talk about mushrooms, we usually discuss them in 2 big groups, ascomycetes and basidiomycetes. These phylums divide the kingdom of fungi into groups that can only be verified by using a microscope. So many of you will be familiar with the concept of the tree of life, which represents the idea that all life is related, just as any part on a tree is physically connected to any other part on the tree. And that different groups of organisms have evolved by continual divergence from lineages from an ancestral stock. When we classify fungi, we're asking how this fungus is related to other fungi in the broader tree of life. When we identify something, we're asking, what is the name of this specimen in front of me? So when we talk about a mushroom, we might use a common name, but what we should use is its taxonomic name or the species name. And a species name consists of 2 words. It's the genus name followed by the species epithet. And there are some rules governing the naming of fungal taxa. So divisions end in mycota, classes end in mycetes, orders end in ales, and families end in ace. And these rules are known as the International Code for the Nomenclature, or ICN. So why Latin? Like, why are all these scientific names for mushrooms in Latin? Well, the reason is largely historic, because Latin was the language of science in 1700s and into the 1800s, when the current system of naming came into existence. A great many taxon names were devised in those years, and by the time Latin lost its role as the common scientific language, it would have been an immense task to change every name. So even if they could change the names, would there even be an agreement on which language to use instead? Probably not. So Latin is a dead language, but new words are often introduced in various non-Latin languages and then Latinized and then used. So a common classification scheme for living organisms uses the concepts of such as species and genus. In this scheme, the species is the basic unit. So a genus typically contains a number of species, and species is said to be of lower taxonomic rank than the genus. Other examples of taxonomic rank are in this classification scheme are family, order, class, phylum, and queendom. I'm going to change that to queendom instead of kingdom, so each of those is a higher rank than the one below it. So this means there are fewer genera than there are species, fewer families than there are genera, and so on. It's clear that this is a hierarchical scheme, and in this system of classification, it's an example of a taxonomic classification. Any particular species, genus, family, and so on is an example of a taxon or taxa. So this mushroom is the Helvella maculata. So ascomycetes are a taxonomic rank in the classification scheme at the phylum level. You can see here, this is the Helvella maculata. It's in the kingdom of fungi, the phylum of Ascomycota, the class of Pizizomycotes, in the order of Pizizales, in the family of Helvellaceae, and the species name contains the last two parts, the genera and the species epithet, the Helvella maculata. The Basidiomycetes are a taxonomic rank, and the classification scheme is at the phylum level. So you can see here the Amanita augusta is in the kingdom fungi, the phylum of Basidiomycota in the class of Agaricomycetes, in the order of Agaricales, in the family of Amanitaceae, and the species name contains the last two parts, the genera and the species epithet, Amanita and Augusta. The interesting thing is that this phylum is broken down using microscopic characteristics. You can't tell just by looking at the macro mushroom in your hand where it belongs in the split. You have to look at how the spores are formed. So in ascomycetes, spores are encased in a sausage or tube structure there called an asci. And you can see there's some little round spores inside. So here's some examples of ascomycetes. This is Rhytisma punctatum. It's the little tiny black dots on this leaf. This is Sarcosphaera coronaria. Plectania milleri. Donadina nigerella. Ernilla pattoniana. Calycina citrina. Gyrometra ambigua. Helvella maculata. And Xylaria hypoxylon. Ascomycetes are my favorite group to study, and I always try and seek them out when I'm on a foray. This phylum of mushrooms does not contain gilled specimens as far as I know of. So these are Basidiomycetes, and they develop spores at the tips of their basidia. There are different shapes of basidia, but this fungi in particular is this orange jelly fungi called Dacromyces chrysospermis, and it has two-pronged basidia, and each tip of those basidia grows and holds one spore each. And this is the spores for that Dacromyces. So here's some examples of Basidiomycetes. These are Boletus edulis, Hydnum pecii, Sterium hirsutum, Astraeus pteridis, Chlorophyllum olivarii, and the Nematelia aurantia or tremella. I still have to figure out where that fits that name change, this name change thing. So Basidiomycetes are generally gilled mushrooms, but they also have pored, tooth, and jelly fungi forms as well. So I mentioned these classifications and the differences because it's a part of the identification process and a key component to discovering what mushrooms will fruit in what season. For example, most of the ascomycetes I come across are in sort of the cooler months, where Pseudotsuga, such a huge phylum, I find them all year long. So now we're going to talk a little bit about how the mushroom grows, the precipitation and habitat that will produce fungi. And you've heard about the climate crisis and how it will affect the seasons. So now, sorry, and I just wanted to say like how we sort of went over how to begin to name some mushrooms. So I want to show you some of the kinds of mushrooms and observations that I have found in each season. It's not an extensive list, but if you want to see more than what I'm going to show in this presentation, you can go to our iNaturalist and find our WVMS funga project, and you can see all of our observations here. And not just mine, but the whole study group here. So I am going to grab a drink of water and give us like a 10-minute intermission. And then we're going to start going over my observations per season. And if anybody has any questions right now, I can take them. Otherwise, you know, just stop looking at a screen for a little while. So let's take 10 minutes. Diana, I can't hear you if you were trying to talk.
I'm not.
Oh, okay.
I'm getting a horrible echo, but it was great so far, Autumn.
Awesome. There's a lot of resources in there. Start recording again. Okay, here we go. Okay. So now I'm gonna show you some stuff that has happened in each season that I've observed. Okay, so now you know a little bit more about how a mushroom grows, the precipitation and habitat that will produce fungi, how the climate crisis will affect the seasons, and how to begin to name a mushroom. Now I want to show you what kind of mushrooms I've found in each season. And this is not an extensive list, but like I said before, go to our iNaturalist if you want to see everything that we found. So winter is December, January, and February. But just remember that these are generic months for this season. And a lot of these mushrooms overlap into other seasons. So I'm going to show you what I've found in the coastal range and the Cascades within a 50-mile radius, or about an hour drive or less from my home in South Salem. So this is the Hydnum umbilicum or Hydnum oregensis. It's a basidiomycete, and it's found in mixed conifer forests, usually in troops or arcs. And you can find them sort of in like clustered lines growing together. They do grow in the same areas as Craterellus tubiformis or the yellowfoot chanterelle. And they are pretty hardy mushrooms. If you've noticed that it's been in the freezing temperatures or snow has fallen, it's not like they're just going to melt away. So, get out there and look. And these are one of the best edibles, in my opinion. So on iNaturalist, there is a wonderful feature to show the seasonality of fungi. They have graphs on here that show seasonality and history and stuff like that. But I just want to sort of make a disclaimer for this, that the graphs, the data that they pull from these graphs are not perfect, because the data is pulled from observations that was submitted to iNaturalist and not when the mushroom was observed. So often people will wait weeks or months to upload their observations, and so it skews the data a little bit. But I've found that generally these graphs are correct. And I often find most of my mushrooms during the peaks in these graphs. So I'm going to pause my presentation again. And we're going to head over to iNaturalist. So I encourage everybody to sign up for an iNaturalist account. It's free. And this has been one of the best things our study group has found. It's sort of the leading way for citizen scientists to help push the study of fungi and plants and all kinds of things. So after you have created an iNaturalist account and logged in, you're going to go up here to Explore. And right here in the species, we're going to type in Hydnum oregensis. Now, this is where those Latin names that I told you about are going to come in handy, because often the common names do not pop up here. So you have to know those Latin names of the mushrooms. So here's Hydnum, and you see it sort of autofills, and you can just click on the species. Okay, so now we're going to go over here to filters. And you're going to go to more filters. And we want to change this to the state of Oregon. So here we go. Select that. And that brings us to the state of Oregon. Click on fungi and update search. So now you can see we only have the observations of the Hydnum for the state of Oregon. So you could do this for, if you're like traveling out of state, you could do this for any state. So you can see on here, there's like each one of these red dots is an observation. There's like maybe 10 observations on here. There isn't that much. We can zoom in a little bit and see this. And you can see they're in a lot of the same places that I mushroom hunt. Okay, so there's a little trick that I want to show with this too. So a lot of people don't know what they have. And when they upload a mushroom, they don't necessarily put in the species. Sometimes they'll just put in the genre. So let's do that now. So we're gonna go here and we're just gonna put in Hydnum instead of the species. And we're gonna select this, the whole genus. And now you can see we get a lot more observations. So if we, let's just click on this one, doesn't matter what, and let's click on the name, Hydnum orgensis. This is where you're gonna find those seasonality charts. As soon as it loads up here, you'll be able to see, and you can see that there's about, 30 observations here, and the peak seems to be in February. And I often find most of my hedgehog mushrooms between, February and March usually is when I find them. So if you go down, scroll down on this page, you're going to see taxonomy. And this is where you'll find the full list of where Hydnum orgensis sits in this classification scheme. So you can see that there's more taxon than just what I had said. Like, so there's subphylums and there's subgenuses, and there's a lot more than what I had suggested earlier. There's even subspecies too. So just be aware that I tried to simplify it. And while we're here on iNaturalist, I'll show you how to search for our project. So if you go to WVMS funga, you will see it here. And you see we have 685 observations right now. There's only 5 of us observing into this, and we've already observed over 230 species. Now, we just opened this iNaturalist together about a year and a month ago. So we've done all that work in 1 year. So I show you this because you can go in here and sort of see where a mushroom grows, and you can see the observations and sort of get an idea on a map where to find these things, okay? But you need to be like a responsible mushroom hunter and make sure that you are going to land that you can hunt on. Make sure that, the observations you look at are just more for general direction, and then you go to the national or county forests and make sure you get the correct permits before you go mushroom hunting. Don't trespass on any private land, and follow all the guidelines on your permits. Okay, back to the presentation. So that's our seasonality. Okay, so this is Plectania milleri. It's a little black cup fungi, one of my favorites to find. It's an ascomycete, and I just wanted to share that Plectania comes from the Greek word plect, meaning plated, twisted, or coiled. And in this Plectania, that refers to the twisted base of the stem that goes into the substrate. So you can find this one in conifer forests. I've found it most often on branches and twigs, sort of clustered in mossy, duffy areas. These are pretty freeze tolerant, and in order to ID these, a microscopy is essential. You really can't tell these black cup fungi apart unless you're doing microscopy. So the seasonality of these, you can see peak in about February. So they're around pretty much all winter. Okay, this is the Astraeus pteridis. This is a basidiomycete. Astraeus comes from the word aster, meaning star, which is why the common name is Earthstar. In this case, it's the false Earthstar. So these are found in conifer and hardwood forests. They're usually you'll just find the one mushroom, or you could find them sort of scattered really far apart though. They fruit on the ground, and when they're older, their rays on that star part start cracking. So they also have like a rounded puff ball in the center. And when they're mature, you can squeeze them and the spores will puff out of them. You can see that these, the seasonality for these is all winter, but they do overlap into fall and spring as well. This is Clavulina rugosa. It's a basidiomycete, and clavulina comes from the Latin word clava, which means club. And lots of these club fungi have that root word in them. So we've mostly found these in Douglas fir forests. They usually are in like troops or clusters, and I think they're probably mycorrhizal, rectomycorrhizal, because they're always fruiting near like the roots of the Douglas fir trees that are buried under the ground. There is a lot of lookalikes to these, but our study group has labeled this our friend, because we see this all the time when we're out mushroom hunting. And we have observed it as early as November, and last year we were still finding it through April. So we see it all the time. It's probably the most observed mushroom that we see.
Autumn?
Yeah.
So that mushroom is different than a Romeria coral?
Yes.
Okay.
Definitely. It's in a whole different genus. Okay, so here is a video.
So I am here in a hemlock forest at about 2,000 feet elevation, and I wanted to show you all how these yellowfoot chanterelles, the winter chanterelles, are growing. So you can see that they grow in some pretty extreme conditions. So here we go, we've got a nice patch of snow. And right here, got a nice winter chanterelle. Put that in the bag. I can show you these here. And they're just gorgeous. They're prime, not soggy or wilty. And they are like all over here. There's just a big troop of them. So, yay, get out in the winter.
So I wanted to show you that because I've noticed quite a few people in the mushroom ID group are a little bit leery about going out in the winter and the rain and the snow, but we have found such amazing things out there in the really snowy, rainy, wintry conditions. So don't be afraid of it. Get out there and do it. So this is the Craterellus tubiformis. It's a basidiomycete. The tubiformis comes from the Latin word tube, which means tube, and form, which means shape. So that's because the stipe of this mushroom is shaped like a hollow tube. This one fruits in conifer forests. In that video, I was in a hemlock forest. They're often found in troops or clusters, like you could see there. They fruit in the needle duff and on mossy stumps. And you can often find them near the hedgehog mushrooms or the Hydnum. Like you saw in the video, they're pretty freeze tolerant. With a light dusting of snow or freezing temperatures, you can still find them. They aren't going to be, too soggy. And the seasonality for these, I have never found them as early as August, but it looks like there's lots of observations then. So maybe other people have seen them, then, but I generally find the Craterellus tubiformis after January. This is the Helvella maculata, and it is found in troops, sort of scattered, sometimes just singly, fruiting in the ground in conifer forests. They have a fluted stipe, and the word, or the prefix macule means spotted. And so this cap has this sort of spotted brownish gray, head to it. And you can see the seasonality here. I have almost found these all year round. And so I've found them as early as September and yeah, into May. Like there isn't hardly any months that I haven't found these guys. They do prefer cooler weather, so. Now this one is a fun one. This is a Spherobolus stellatus. This is a basidiomycete, and these are really interesting little mushrooms. From what I read, they're in the same family as the Earthstars, but in David Aurora's book, Mushrooms Demystified, he had them in the same area as bird's nest fungi because they're so small. These are found in conifer forests, and they're in huge troops on conifer branches. And they shoot out the spores in a little tiny package called a peridial. So if you've ever seen a bird's nest fungi, you've seen the little tiny eggs inside. This has one little set of eggs, and this mushroom will create that bubble and shoot them out like a cannon. So it's kind of interesting though, because this sphere like throwing mechanism on this mushroom needs light to trigger that shooting effect. It can't be in complete darkness. And you can see the seasonality of these are almost all year round. I've found them in late winter and early spring before. This is Pseudohydnum gelatinosum. This is a basidiomycete. Its common name is called the cat's tongue. I find these in conifer forest pretty much as I find the Clavulana rugosa. It's all over the place. These are usually found in scattered troops on decaying conifer branches and logs, or sometimes really rich decaying soil on the ground. The hymenium surface is covered in little tiny spines, and these are sort of a translucent, they can be white to brown like you can see here, and these are edible. We find these pretty much from August to March, and I can concur with that. I find them almost every time I go out. All right, this is Xylaria hypoxylon. It's an ascomycete. Its common name is also called candle snuff fungus. You can find it in mixed hardwood forests. they are usually found in these little troops or clusters and fruiting on decaying hardwood branches and hardwood roots. If you see these, the wood could also be buried, so make sure you dig around them and check it out. So the white tips, like you can see here on this one, those are actual asexual spores, where the regular spores that you would want to identify the mushroom are here on these little raised spots in this actual black part of the fungus. These are excellent decomposers. And we find these anywhere from October all the way through spring. But you can find, dried, like old specimens of this all year too. And this is Astrascalpium vulgare. This is a basidiomycete, and the Latin word scalpium means pick, and thus the common name of earpick fungus. So this is the mushroom I told you I would share with you earlier, and it is fruiting on a Douglas fir cone. They love these things. You'll often find the mushroom just solitary or with an older specimen. And these cones are usually buried. So if you find this mushroom, dig down and you'll find the cone it's fruiting off of. This has a toothed hymeneum surface, and it is excellent camouflage. Jordan was the first one to find it this year and kept finding it and making us all jealous that he was finding it. I finally started finding it, so. Okay. And you can find this in from about October, after the first rains come, all the way into April and May. Just sort of depends on how moist that forest stays. Okay, let's talk about spring. This is, March, April, May-ish. All right, this is Traumates versicolor. It is a basidiomycete. Its common name is turkey tail. It's often found in hardwood forests or growing just like on some hardwood. It is a zoned polypore mushroom in browns and cream colors. And the important identifying feature is to take one of those mushrooms and flip it over, because the pored side will be white. You can see that the seasonality for these are pretty much all year, although some of those observations were probably really old mushrooms. And so we generally find these in starting about maybe November at the earliest is when I sort of see them. And they're pretty prolific right now. Okay, this is Sterium hirsutum. This is also a basidiomycete. This is known as the false turkey tail, and you can find it in mixed woods. It's found in clusters, sometimes just, solitary, but I've really seen it just take over an entire log. Fruits on hardwood logs, and it's also fan-shaped, but this one is way more orange or yellow. Especially on that pored surface, it's going to be bright yellow. So you can see to the left here is the cap of this mushroom, and then over there to the right, that one is the pored surface, and it's very yellow, yellowy orange. Okay, this is Donadina nigerella. It's an ascomycete, and this one is found in conifer forests, has a little stipe on it. It fruits on conifer sticks that are often buried, so you got to dig. It has a sort of a shallow cup and elliptical spores. And you find these in spring, usually starting around March and into June. So I'm here in my studio today, and I'm going to show you how to do a section of an ascomycete fungi and look at the spores and asci under the microscope. So today we're going to look at this little black cup fungi. It has a little stipe on it, and we're going to do a section of that. Looking in the ascomycete fungi of North America by Michael Bugh. And I've seen these enough now to know what they are. So I've identified it as what Michael Bugh refers to in this book as Plectania nanfindeltii, which the name has changed now, I think officially, to Donadina nigerella. So we're going to just double check that this is what I have. And to do that, we have to look at some of the microscopic features. So we're going to look for elliptic spores and asci with 8 of those spores in it. So first thing we're going to need is a razor blade to take the section, a spot plate to help us gather those sections so we can get the best one. It helps if you have a pair of forceps to grab those little sections that you make. And then you need a microscope slide for your microscope and a little cover slip to go on top. To mount your slide, you're going to need some distilled water or some reagents. Okay, so in this mushroom, we have the hymeneum surface. And on this one, that is the inside of the cup. That is where the spores will come out of. And then the abhymeneum surface, which is the outside of this. And there won't be any spores on this side. So when you take a section of the mushroom, you want to make sure that you're going to see the spores. And so it'll be on the inside of this cup. So how we're going to do that is we're going to take a little section off of this mushroom, like this. So you can see I've just cut out a little piece all the way through the mushroom. So I have some of the hymeneum surface, some of the abhymeneum surface, and then some of the flesh as well. This surface on this side here is the hymeneum surface. And if you flip this little piece over that I cut, there we go, that's the abhymeneum surface. So I'm going to try and get a really thin section cutting this way. So I've loaded up my spot plate with some DI water so the material can float in there. And now I'll try and get a super thin okay. There's a really thin section, but it's actually still too thick.
So I'm going to try again. And that is looking like a good one there. So I'm just going to float it in here while I get some more. So there's another section, kind of thin, but I think I can go thinner. The thinner you go, the better, the easier it'll be for you to see under the microscope. So in here are some little sections, and I'm just going to put them on. With a drop of water and get another one of these. So then we take our cover slip and you just lay it on top. So I like to squash mount my stuff. So that means I just press on it a little bit, try and get the air bubbles out. My sections aren't thin enough to be perfect, so my cover slip moves around a little bit. Okay, so now this slide is some little sections on it. You can see the cover slip on there. There's water in between the cover slip and the slide. Now this is ready to go in the microscope. I'm at my microscope now, and I've got an awesome little camera that goes in my microscope lens so that I can see what I'm doing on my computer screen. So I'm just gonna hook all of this up right now so I can see what's going on. I have my microscope set up at like times 100. So it's a good place to start for me so I can sort of see exactly where we are on the slide. Okay. So here's the slide that I just made. I'm put it in my microscope here. So now I'm going to adjust my microscope and get an idea of where we are on it. Okay, I'm starting to see some stuff on here besides air bubbles. So I'm going to focus in. Okay, I see some asci and parentheses and spores. And as I zoom in, they get focused. The spores are elliptical, which is right in line with what the Donadina nigella is. So you would want to read through the microscopic features that's listed in your books and just sort of check things off. And if they match up, then you would have the mushroom that's showing in this book. So let's look. Okay, so now I'm going to move up to a times 400 so I can really see these spores. And I need to adjust all the lighting and stuff so that I can see it. They are elliptical, about a 2-to-1 ratio, elliptical. And they do have little oil droplets in them. And let's see, the paraphyses are abundant, it says, and there's tons of them in here. So that is how you would look at something under the microscope.
Okay, so this next section is springtime in a burn area. And for this part, I did travel onto the eastern side of the Cascades. So this is the only mushroom hunting I've done over there. This mushroom is the Sarcosphaera coronaria. The Greek, this one is an ascomycete, and the Greek root sark means fleshy. And the word coronary comes from the Latin word corona, or Greek word koron, meaning crown. So this is a fleshy crown. These contain a high level of arsenic and should not be consumed. So they're found in conifer forests. I was finding them under pine forests, in clusters or just single. They fruit in the ground and they are large purple cup. I mean, these things are probably, I've seen them up to about 4 or 5 inches wide. They have a sort of star-shaped margin, and these are a good morel indicator. So if you are seeing these large purple cup fungi, look around for morels. You can see the seasonality of these mushrooms are pretty much just in May. And that's when I found the specimens that I did last year was in May and June. This is Pyronema omphalioides. This is also an ascomycete. The prefix pyre in Greek means fire. And this, and the root omphal in Greek means navel. So if we look at the fruiting body, we'll probably see some sort of a navel-like depression in it. But this is something that you'll find in burned forests, and you find them about 2 weeks after a burn, although I did find this the season after a burn. So, you know, I don't know exactly why we were seeing it so late after that burn, but it's a matted cluster of really bright orange fungi. And as it gets older, it develops these sort of orange cups. And you can see the seasonality of this is in April, May when I was seeing it, but people have also observed it in October and November too. Just must be, you know, after the burns is when people are finding it. Okay, this is Gyromitra esculenta. It's an ascomycete, and gyre in Greek means round. So the Gyromitra means round hat. Esculenta in Latin means fit to eat, but that's a deceiving name for this mushroom because this mushroom is actually very toxic. This mushroom contains the toxin gyrometrin, or monomethylhydrazine, MMH, which is the same stuff as rocket fuel. So I would not recommend eating this. This one is found in conifer forests. It's usually in scattered troops, fruiting in the ground. It's got a brain-like cap, and it's also known as false morels. The seasonality for these are right around the same time morels are fruiting, and also the sarcospheric cornarias, the purple cup fungi. All of this stuff is, all these ascomycetes are always found in spring. And the morchella. These are the ascomycetes. I don't know as much as Jordan does about morels, and so I'm not sure what species we were finding. It's very hard for me to figure out the species in morels because they can change their color and shape and way they're growing and stuff from mushroom to mushroom. So I have a lot more to learn on that one, but you can find morels in burned areas. That's where I found these. They're found in scattered troops, fruiting on the ground. They have this like pitted cap instead of the brain-like cap, like the Gyrometras. And the stipe and the cap are attached, where the Gyrometra's cap is usually pulled away from the stipe. And these are edible and they're delicious. They're probably my favorite mushrooms. And like Jordan had said when we were on our break, seasonality, yes. April, May, I found these the end of April and May last year. So we're looking forward to a good morel season this year. Let's hope. And like Jordan said, here's our Boletus rexvaris. These were some amazing boletes that we found in the burned areas last year. These are spring boletes, and they're found in pine forests, usually in scattered troops. You find them in the ground. They have this pinkish brown cap and a bright yellow pored surface. You should look for the fishnet reticulation on the stipe there. Often though, these mushrooms don't really pop out of the ground. You'll find them more as mushrumps, which are just like humps of dirt that you have to pull the dirt away to find the mushroom. If you find these mushrooms, sticking out of the dirt, they're often too buggy to eat. So look for the mushrumps. And these you'll find in the same time frame as morels, and it looks like even a little later. Okay, so I'm going to take you through how to find where to go morel hunting. So this is the Bureau of Land Management, or BLM website, and all of these are on that sheet of resources I gave you as links. So this is blm.gov. And if you scroll down on this website, you're going to see maps here. So let's just click on the Holiday Farm fire map. And you will get this nice map here. And it's a PDF. You can download it and save it, figure out where the burns are. You can do that with all of our more recent fires, the Beachy Creek and Riverside, and these Archer Creek fire here. But now I want to go down here to the Forest Service fire closures. And this brings you to the USDA.gov for our Pacific region, and it's basically the website for our national forests. So if you go here, we're going to go ahead and click on Willamette National Forest. This is the one I mostly hunted in and was burned really, really bad. And you can see here, if you just read, that the following places are closed. So nobody is allowed in any of these places at all. And Marion County specifically sent out a press release the other day, and I sent it to our members, but they're fining people taking vehicles, you know, and even doing jail time for people they catch out in here. So please don't go, especially to the Willamette National Forest, until you see, like, check this website before you go out. So let's click on the Beachy Creek and Lions Head Fire map here so you can see it. And this is also a map that you can download. So back on this, and there's the other Holiday Fire Farm map as well. So on the sidebar here, you're going to see this thing that says passes and permits. And you can go down to mushrooms right here under the special forest permit. And this is for the Willamette National Forest. And you can read through all this about what it takes to get a mushroom permit. And you would need to do this for every single forest that you might go into. So if you look on iNaturalist and find an area where you see lots of hedgehogs happening and you want to go, then take that information and find out what forest it's in, and then make sure you go to this place and find the proper permits and you know, accesses to that forest, BLM land, whatever. All those links are in that document. Okay, so let's move on to summer. So summer is June, July, and August. And summer is one of my favorite seasons for finding mycoheterotrophs. So this is the Monotropa uniflora, and it is associated with the Rusula mycelium, which the Rusula is a basidiomycete. So these are found in mixed conifer forests, and you usually find them in large troops in June, July, August. You'll see them in this like ghostly white color. Sometimes you'll see them in pink and red in their little flowers, but eventually you see them completely all turn black. I start noticing these things a lot in July, and I have observed them all the way through September. This is Hematomes congestum. This is one of my new discoveries this past year. This is another microheterotroph, also called the gnome plant, and it's found in conifer forests. I don't know what fungi it's associated with, but it is found in these sort of clusters, and as it ages, it turns brown. So you'll find these in June, July. We started observing them in like July and August, so keep your eye out for those.
Autumn just found this super cool thing here. This is one of the mycoheterotrophs, and it's related, like, it's not related to Indian pipes, but it's in that same sort of a group, where the microheterotrophs are those that are connected to fungus underground, and in turn, the fungus, the mycorrhizae, the hyphae, are connected to tree roots. So basically, this is a little plant that has no foliage for chlorophyll, so it gets its nutrition ultimately from the trees that are around it. It's super cool, and I actually can't remember the name of this one right now. It's Hemi something.
We'll put it on the video later.
Yeah, but this is a really cool discovery. I've never seen this one before. I'm very excited.
So point out the plant parts again.
Okay, a little botany lesson here.
Yeah.
This is actually a cluster of flowers. And this little brown point right there is the stigma. And the stigma is what catches the pollen. And the pollen, let's see, I don't know if we can find it, on the stamens. I don't want to pull these apart too much. If we can even get to it. There we can see them down there. They're these tiny, tiny little thready things.
Yes,
That's what holds the pollen. And these are little remnants of petals. And this one over here, if you look at this one, you can see that this is starting to swell up a little as the ovary, and that will ultimately be seeds. And look at, there's another one.
I know, I saw that one like at the base of this tree trunk.
I know it. And these are shore pines.
And they're shore pines.
Very nice. Oh, here, I'm going to picture this. This is another, it's not, this is probably a mycoheterotroph. I have to do a little research, but this is Gutiera, unless they've changed the name. They call it rattlesnake orchid or rattlesnake plantain, but it is technically an orchid. There's tons of them in here, and this is a flower stem which has not opened quite yet. But there's lots and lots of these in this forest, and I'm surprised to see them.
So interesting.
I know.
All right, thank you.
It's all connected.
So this is another summertime mushroom that we were finding. It is the Tapenella atritomentosa. It fruits in conifer forests, grows scattered or solitary. We were finding like a couple fruiting at a time. They are a really slow growing mushroom. It took almost 6 weeks for one of these to really fruit out to that pancake size. They have a really velvety cap and stipe, and the gills are forked and decurrent. You'll find these in sort of peaking in August, and we found it from August and observed this thing all the way through October. This is Hydnum suaviolens. It's in a conifer forest. This one is amazing and smells like anise. You can find it under Sitka spruce, and it's got this funnel shape with a really pretty blue stipe. The seasonality of these are summertime, like August, September. We were finding them in August. This is Hydnum peckii. It's the bleeding mushroom, or strawberries and cream. It's found in conifer forests, usually under pine and spruce, and it usually find it fruiting on the ground. It bleeds when it's young, and it's really velvety and pink and really big when it's old. Now these are poisonous, so be careful. The seasonality of these are in August, September, like so from summer into fall, and I have found them in August and also in late fall as well. This is Hypomyces lactiflorum, or the lobster mushroom, and I wanted to show you the different stages that this mushroom goes through. So the white is the beginning of the parasite of the Hypomyces attacking the Rusula, its host. And you can see over on the left, it is like a light orange. And then over on the right, it's gets really, really dark. It can even get like dark purple red as the Hypomyces take over the host. These are found in conifer forests. Our mushroom club found tons of these at Lobster Palooza a couple years ago when we camped. It was a lot of fun. They're delicious, edible. We can find these in the end of summer and into fall. So start looking for them in about the end of July and into August. This is Amanita augusta. This is a basidiomycete, and this specimen was just so stunning. I found this in a conifer forest, just the single one. It fruits in soil and has that brown spotted cap and this yellowish stipe with the vulva and annulus. And you can find those from late summer and all the way through winter. Now we're going to go into our last season, autumn. And this is September, October, and November. This is the Rusula nigricans group. It's a basidiomycete. And the Rusula nigricans grows in conifer forests. This one will stain like this wine red when you handle it or cut it or bruise it. And then it turns black when it's old. The seasonality for these is about October to November. I've seen them as early as, September though, like right after the first rains. So they get really black in October though. And this is a fun thing that grows on those black russulas called Astrophora parasitica. And it's a basidiomycete, and it's like the Hypomyces lactiflorum. It's a fungus that grows on another fungus. You can find those anywhere you can find their Russula blackened hosts. These little guys have whitish, brownish caps. They're gilled, and you find them in large troops. They almost look like pinheads in a pin cushion. They're really, really cool. You can find those. I found mine more in like October. I never have seen them at the end of summer, so I don't know about that. I really think they need that blackened Russula to for its host. So, like I said about these observations, these graphs, we'll see. The chanterelle. This is Cantharellus proliformosus, although I'm not exactly sure. I've never actually had time to microscope these and see the spores because I just get them home and eat them so fast. These are basidiomycetes, or the golden chanterelles. There is a lot of work being done on genetically testing this whole line of chanterelles because they really think that there's more species here than we think there are. So these are found in conifer forests, in clusters or troops, fruiting in the duff. They've got the golden yellow caps and blunt decurrent gills, and they are delicious. You can find these if you've had a really good rain as early as late September, but I often don't find these till like mid-October or beyond. And then once the cold snap comes in, they're done. They just turn to mush as soon as it freezes. These are the Turbinellus flaccosus. They're a basidiomycete. The word turbinellus comes from the Latin word turb, meaning something that whirls, like a top. And in this case, the mushroom is cone-shaped. Flaccosus comes from the Latin word flock, meaning a tuft of wool, which is why it has its common name, the woolly chanterelle. This is not a chanterelle, though. This is a false lookalike. You can find it in conifer forests in clustered troops. It fruits in the duff. It has a really bright orange cap, but sometimes they fade to that sort of chanterelle yellow, and they also have blunt and decurrent gills. If you eat these, though, they can cause nausea and vomiting, so be careful. They are found generally the same time golden chanterelles are found. I usually see them in September, October. This is Crugomphis tomentosus. It's a basidiomycete, and you can find these in conifer forests, basically the same sort of habitat, and they have the same sort of look as far as the coloring goes with chanterelles. These, though, have widely spaced gills. They're not blunt gills like chanterelles, and these have a veil when they're young. Again, you can find these in this exact same season that you can find chanterelles and the Turbinellus flaccosus. And you can see here, my kids were on a foray with me, and they had put all of these in their basket thinking they were all chanterelles. And so just a word of caution, the two there on the left are chanterelles and the rest are not. This is the Amanita muscaria. It's a basidiomycete. This is your classic fly agaric, found in conifer forest or sometimes in wood chips. It's usually just one, but sometimes you can find a little cluster of different ages, like I found there. They have bright red warty cap, a partial veil, and a vulva. You can find these pretty much as soon as it starts to rain until it gets cold. So I've seen them from August through December, really. This is the Lacaria amethystio occidentalis. It's a basidiomycete, and it is like this fluorescent bright purple that my camera can't even capture. The color is just amazing. They're called the Western Amethyst Lacaria. You can find them all over the conifer and hardwood forest, especially in Douglas fir. They're often found in really large troops. They're bright, bright purple, this bright fluorescent purple when they're young, but they turn to this like pinkish, purplish, brownish color when they're old. They have those widely spaced gills. And you can see they're most common in January, although I have found them from November into January. This is the Gomphidius oregensis. It's a basidiomycete. It's usually found fruiting near or with Swillus species. You can find them fruiting in the ground in a Douglas fir forests. They start off with this really cool yellow stipe and sort of a black or a brownish, grayish, really slimy cap, but they get really black when they get old. They're kind of interesting mushroom. You can find these in the fall and into December. I've found most of mine in October and November. This is the Boletus edulis, and it's a basidiomycete. These are our fall boletes, the king boletes. Find them in conifer forests, usually found in scattered arcs, often, again, fruiting in those mushrumps. So look for the tufts of dirt that are just humping up. The caps on these are sort of an ochre tan color to warm brown, and they have a really thick stipe in a lighter color than the cap. You can find these at the end of summer and into fall. I've found most of mine in October. This is Lepiota. I'm not sure what species this was. I didn't really know that much about them when I started, but this is a basidiomycete, and all Lepiotas are poisonous. So they're really pretty. They're smallish, like 5-6 inches and under. They they can be in urban settings or beside roadways. You find them in small troops fruiting in the duff. They have a domed cap with this dark spot in the center, and they have a partial veil and all these like clumped fibrils on their caps. So one of the species has those fibrils on its stipe too. And you can find these mostly at the beginning of the mushroom season. I was mostly finding mine in October. This is the Chlorophyllum olivarii, and it's a basidiomycete. I wanted to show this one too because it looked similar to the Lepiota, except these are huge. These are about 11 inches tall, and those caps are almost 5 inches wide in diameter. So you can find these in disturbed forests. They're usually found in troops or arcs, fruiting in the duff. They have these sort of fibrils and like clumped hairs on the caps. They're really pretty. They're a really large mushroom, and these are not poisonous. And you can find these in the fall, October, November. I was finding these in late September and October. This is Thelliflora palmata. It's called the stinking earth fan, and it's found in mixed forests, usually in clumps. It's often overlooked because it just blends right in with the path you're walking on. But if you ever find one, pick it up and smell it. It smells disgusting. It smells like rotting cabbage. And you can find these almost all year round. Generally, though, I've found them in October and November. This is Lactarius rubidus. It's a basidiomycete. It's our candy cap mushroom. You can find those in mixed forests, especially Douglas fir. They're usually found in large troops fruiting on decaying stumps, Douglas fir stumps. They have a rough cap with this watery white latex. And they smell like maple syrup. They do have lots of lookalikes, so be careful. You can mostly find these in fall and into winter. I have found them a lot in November and December. This is the Marasmius plicatus. It's a basidiomycete, and this is one that we learned this past fall, and we then saw it everywhere. So this one is in a mixed forest, especially in spruce. Found in sort of scattered groups in duff or leaf litter. It has a velvety reddish brown cap with creamy white, widely spaced gills, and it's on this thin, wiry stipe that's pretty hardy. It doesn't really just break. So it's about 5 inches tall. So next time you're out in the fall, try and find this one. They come out about with the first rains, so October, but you can find them through March. We were finding them often from October to March. This is Calocera viscosa. It's the yellow tuning fork. There's lots of yellow club fungi in the forest growing off logs. This one grows off of conifer logs. It's often found in these clusters. They have this forked tip at the end of their club, and they're usually bright yellow, but they do have a lot of lookalikes. You can find these in the fall and all the way into spring. And this is the last mushroom I'm going to tell you about. This is the Tectella. This Tectella is a mitopic genus, or a genus that has only one species, the Tectella pteridis. And according to the paper written off of this European species in 2012, they're in the Mycenaceae family. This species is rare, with only a few observations of it on iNaturalist. And I found it. I found it this past fall, and I have sent a sample to get genetically tested and sequenced, and I'm just waiting on the results. The description in that scientific paper wasn't exactly what I had here, so I may have found at the maybe another species to this genre, or if not that, I can at least help broaden the description of this mushroom. So I'm really excited, and I'll keep you all posted. This one was found on alder in a hardwood forest. This particular one was growing in these different aged clusters. It was really, really, really slimy. Like, you could barely hold on to it. It had this soft yellow color and these tiered gills underneath. And you can see here, just from the observations of it, like, there's only been 3 observations of this mushroom, one of which was mine. So I just want to say thank you to WVMS Study Group for the wonderful year of fantastic fungi finds, and I always willing to explore and listen to my bad Latin as I learned fungi. A huge thank you to my mentor, Henry Young, for always pushing me to find the answer. And even though I've been wrong more than I've been right, thanks to Diana for helping me understand habitat and connections to fungi and making time to crawl through the forest floor with me. Thanks to Jordan for patiently answering all of my questions and helping me find places to explore and generally geeking out over that next mushroom. And thanks to Jeannie for sharing your incredible lichen knowledge and pulling out all the old college collections and textbooks and always being willing to participate and learn more. Thanks to my family, who's always listened to me talk about mushrooms and go on forays with me, and finally to WVMS for giving me the opportunity to create this study group that I find a challenging new path to learn on. And that's it. I'll take questions.
Thank you. That's a lot of work.
You're welcome.
Hey, good job. That was awesome.
Yay.
Does anybody have any questions?
So I just have to say, you've done an amazing amount of work, all of you, in the last year, and it's really impressive.
Yes.
It really, really is. And I think the other thing that I see is so incredible is how you've linked to the national scene for what you're doing and the amount of knowledge that's out there. And shoot, I wish I didn't have to go to work so I could go out with you guys on like non-end of the week days. This is great.
Good. Yeah, we're hoping that people come to the, Monday night ID sessions, because it's sort of an extension of our study group, and we can talk about all this stuff, not that many things that I just presented tonight, but yeah.
Autumn, can you talk about you said there's a new study club group forming? Can you tell us about that?
So as I was learning and forming the study group, I was also developing a curriculum for future study groups. And so all of our WVMS members are welcome to form a study group and go through the modules that I have created. It's sort of a self-paced curriculum. So we had a member email our group last week and ask for people to be members in a new study group with her. And so as she forms, I'm just sort of facilitating her to start the modules that this group has created. And so we're going to just sort of hold their hands, this next group, and teach them all the things that we have learned. And so, yeah, we're excited. We're excited to make this beyond us now. And it'd be amazing if, more members would be interested in this. Like, you don't have to, if all you're interested in is edibles, then just go through that modules and learn about the edibles. You don't have to learn about everything like this group has done. If your interest is in dyeing or doing something else with mushrooms, then you can definitely utilize the modules to focus on just learning about that. But yeah, Henry's been amazing with helping with all this, and he sets me straight all the time. But I feel like I've done a pretty good job of learning a lot in the past year or so.
I'll say.
Yeah, I know. Yeah. I've only been doing this for a year.
Yeah, Henry's been a blessing. He's pointed us in the right direction many a time when we've been going down the wrong path, especially
I know.
When you get into down some rabbit holes researching certain types of fungi. Oh my goodness.
Yes. And he's the one that has really helped, push this group into doing things outside of WVMS, like becoming part of the Fundus project. And, and so he'll just like tell me about these things, and then I'll go sign us up for it. And I'm like, okay, now we're a part of this thing. So we're just trying to take every opportunity that we can right now.
So is this presentation going to be available on YouTube?
Yes. So